Hello and welcome to After the Dig, the archaeology podcast for everyone who loves digging up the past. I'm your host, Crystal Miller. I'm an undergraduate in bicultural archaeology, scientific communicator, and D&D Dungeon Master. Here I'm joined by early career colleagues to discover what happens behind the scenes after the digging is done. Today we have a bone to pick, and to be honest, it's going to be an osteoblast. Joining me today is Mia van den Dolder. Mia's also a bicultural archaeology undergraduate at MQ. Hey Mia, welcome to the show.
Hey Crystal, thanks for having me, and thanks for that humorous opening.
I'll see myself out. But before I do, let's have a quick chat about a shared passion of ours, bones. First of all, tell our audience just a little bit about yourself.
Yeah, absolutely. So I'm currently completing a Bachelor of Archaeology with a major in human biology and a concentration in health and disease. And one of the main things I'm passionate about is pathology. So I'm currently hoping to pursue a career in anatomical pathology.
What a degree. And what about in your spare time? What do you get up to?
In my spare time, I like to read and, as you know, play D&D.
It's true. She plays in my table. She plays a tiefling ranger barbarian called Persephone.
Absolutely.
Moving on from D&D, tell me, what's your favorite thing about studying the past?
I think my favorite thing about studying the past, especially from the perspective of analysing human remains, is the ability to humanize skeletal remains and take them from just an object to be studied to someone who was alive, and to then reimagine what their life was like based on scientific fact.
Yeah, I think that's one of the things I love about the past too, specifically human remains, right? You can read them like a book, and for the most part, they provide an accurate account of someone's life, almost like a memory. Now, that being said, you have a slightly different perspective from me, a microscopic perspective. Did you want to tell our audience about your favorite method of studying the past?
Yeah, my favorite method of studying the past is looking at bone histology. So this is from more of a pathological perspective rather than that osteological perspective.
So what exactly is bone histology?
So bone histology, basically we take a sample of bone, usually it's a couple of centimeters wide, and then we encase it in an epoxy resin and slice it really thin. And then you're going to use a stain so that you can see the little cells inside the bones. This is called a Schmoll stain. And it stains these bone cells sort of like a blue-black, and then the bony matrix, which is the bone that forms around the cells, that's going to become a yellowish colour. And so from this, then we can tell lots of different things about a person. Mainly we can look at determinations of age or how they died, their diet and lifestyle.
Aha, so while I look at the bones on a gross anatomy level, you're looking right down into the bone to inspect the cells.
Yeah.
After you create a thin slice of bone, dye it, and prepare the slide, what is it that you actually see when you look into the microscope?
I'll explain a little bit about histology and bone anatomy itself. So inside the bone, you have a couple of different cells. So you have three different main types of cells. You have your osteoblasts, which they're your young cells. They're located in the growing bone, and they're responsible for bone formation. So yeah, they're the builders of the bony matrix that you see. And then you have the osteoclasts. So these are responsible for bone reabsorption. So when we look at bone, it's constantly regenerating because it stores calcium for the body, amongst other things. So these osteoclasts, when they're given a message by your body, they then start reabsorbing the bone so they can use that nutrients again. That's how your bones keep on regenerating. And then our final main cell that we look at is the osteocyte. So these are matured osteoblasts, and they're within the bony matrix, and they maintain the bones themselves. So once the osteoblast is matured, it stops creating the bone and it just starts maintaining it.
And they're the most common type of cell in bone, right?
Yes, that's correct.
So when you're looking at bone under the microscope, are you looking at the soft, spongy bone on the inside, or are you looking at the hard, compact bone that surrounds it? Maybe you could explain to our audience the difference between the two?
Yeah, so there is, you got your spongy bone and your compact bone. So we're mostly looking at compact bone here because that's where you get the specific formations where you can have a look at growth and development and things like this. Your spongy bone is where your bone marrow sort of is, that area where they're creating red blood cells and stuff like that. So because the spongy bone doesn't have as much structure that you can predict, it's a lot harder to look at this kind of thing.
So it's that compact bone that surrounds the outside and gives that mineral density and structural support to the skeleton.
Yeah, and there's definitely very specific structures. So within this compact bone, you're going to have perversion canals, which are these canals within the bone. That's where your blood vessels are going to go. That's how the blood is supplied to the bone so that live, because it is a tissue. And then surrounding these perversion canals, you're going to have your osteocytes. So in concentric circles around the perversion canals, they're going to sit in little circles, and then they're going to communicate with each other to help maintain the bone. So one of these they form little columns in these concentric circles, and these circles are called osteons.
So it's within these concentric circles of bone matrix, the lamellae, if we want to get technical, that we find the osteocytes,
Yes,
right?
That's correct. And these can change depending on growth. So in younger people, you'll see a lot more osteons and a lot more unfinished osteons. And then as people mature, they get bigger and they change and they start overlapping. And this is how we can then tell someone's age, or places where maybe they've overworked themselves for a long period of time. You'll see more blood supply, bigger perversion canals and bigger osteons. Or in the opposite sort of situation where they are malnourished or something, you may see like a lack of bone growth. So it's these differences in the bone structure that mean we can then look at what might have happened while someone was alive.
The first thing that pops to mind when you explain how an osteon looks under a histology slide is dendrochronology. It reminds me of, when you slice through a tree and there's all the rings.
Yeah, many little ones all kind of compacted together and overlapping. Yeah.
That's so cool. So by looking at the different cell types, you said that you can learn a lot about someone's life. What exactly is it that you're looking for, and how do these changes in cell structure give you a window into a person's life history?
Yeah, so like I've mentioned before, there are a couple of different ways. One where this is particularly useful is the perversion canals, like I mentioned. So there was a study done by two scholars, Mishkovitz and Mahoney, and this was a case study where they compared two different cemeteries, and these cemeteries were in different locations within the same area. So they're looking at the same population, but they were trying to find different social classes. And so They took bone histological samples from a range of different skeletons over the two cemeteries, and they compared these perversion canals. And then from this study, these scholars were able to determine that one of the cemeteries, they had a lot larger perversion canals, and there were signs of malnutrition as well, which showed that these people were probably of a lower social class, probably like a more working class. They're doing a lot more labor. And what they found very specifically when looking at these bones is there was evidence of labor from a very young age. So they're looking at this kind of juvenile lifestyle.
Laborious and hard.
Yeah, where you're looking at bone development and this enlarged perversion canals from very early on in the life, which was also gave pointers towards this.
And what about the other cemetery? What did they discover there?
Yeah, so they were a lot more standard. The perversion canals were quite either like a medium or even sometimes smaller, and the osteons were just kind of like a general size. There wasn't a lot of differentiation, I suppose.
So basically not a lot of evidence to suggest that people buried in the second cemetery were exposed to laborious or difficult lifestyles during early development. Yep. And I guess that sort of suggests that the demographics buried in each cemetery were different, right?
Yeah, absolutely.
Because the pathological data absolutely points towards a bit of stratification within society there. Now, another good indicator for class differences and stratification within a population is by health and disease markers. What can bone histology tell us about that?
Yeah, absolutely. So disease can show up in the bone in a lot of different ways. Obviously, you have diseases that impact the bone directly, so like leukemia, or like if you have something like joint ankylosing or something that is impacting the bone. But then there are a lot of other diseases like syphilis or hyperparathyroidism, things like that, where it may be like in the case of hyperparathyroidism, which is where your parathyroid glands are overactive, you're going to see the release of calcium into the blood. So these osteoclasts that we talked about before, which then reabsorb the bone, they're going to become overactive. And then this is going to exacerbate situations like osteoporosis or affect the bone that way. So
affecting the integrity of the bone via like a secondary symptom. Now, syphilis is an interesting one. It's incredible because when we think of syphilis, we think of painful looking sores and other symptoms associated with the skin. And to catch everyone up at home, syphilis is a bacterial infection spread by sexual contact and was really prevalent before the discovery of penicillin in the 1920s. The first recorded large-scale outbreak was in the late 1400s, and according to a recent 2020 study, more than 20% of individuals aged 15 to 34 were being treated for the disease in late 18th century London. It's a remarkable disease in its effects on the human body. In primary syphilis, it starts out as nothing more than a sore where the bacteria entered the body about 3 weeks post-exposure. But generally, they're painless and usually heal after some time, so many people wouldn't have even known they were infected. But it comes back, and it comes back as secondary syphilis, which presents as more of a rash and can happen weeks after the initial infection. It's usually not that itchy, but it can produce wart-like sores and have other symptoms like hair loss, muscle pain, and fever. And these symptoms will eventually appear to go away on their own, and we call this latent syphilis because while they appear to be better, they are still carrying the infection, and it can get worse years after the last bout of symptoms. If left untreated, it can evolve into its final stage called tertiary syphilis, and about 30% of people will go on to experience complications such as damage to brain tissue, nerves, eyes, blood vessels, liver, and it can even spread to bones and joints, causing osteomyelitis, which is inflammation and swelling of the bone. And this can cause osteolytic lesions or a dissolution of the bone. An excavation of a 17th century cemetery at St Thomas's Hospital in London found that 13% of skeletons showed evidence of bone damage due to syphilis, which in the grand scheme of things is a small percentage of cases that were found there, which suggests that the hospital was treating patients there. They were likely being treated with the most common treatment of the period, mercury, which, as we can all assume, had a series of devastating effects on already very weak bodies.
Yeah, no, it definitely has an effect as well. Definitely in the skulls you probably might have seen. I think because a lot of it's in the bloodstream as well, and the blood definitely flows through the bones as well. So this affects the structure definitely, especially in syphilis. And this can then be applied to the archaeological record as well. I mean, there's was a study undertaken in the Philippines with skeletal remains from the metal period. And like I mentioned before, the joint ankylosing can be seen in like bone histology. So this study looked at a population size, and they looked at specifically joint ankylosing because they found it in a couple of skeletal remains. And you may have seen this in where they start calcifying the joints. So you can see it from a macroscopic perspective if you're doing something like osteology where you're creating a profile of the bones, but you can also see it histologically, and you can see it before it's evident in like a more major scale.
And I suppose the benefit there is that if you don't have a complete skeleton and you've only got fragments of bone, you can still unearth this information. But tell me, what size of a sample do you require for this? Because if you're taking it from a complete skeleton, then I guess that is quite destructive in a way.
Yeah, absolutely. It's definitely something to be mindful of because this is an invasive technique. You have to weigh the pros and cons. Bone histology is often used for either smaller pieces of bone or fragments of bone. I think because the ethical ramifications are not as great. A lot of the time in archaeological excavations, if you have small fragments of bone or something like that, because you can't really tell as much from the surface, they're often not cared for as much, or they're not looked at enough with bone histology because you need only like a small sample. You can then still look at fragments of bone and then still try and find out about someone. There was a case study done with the Forensic Institute in the Netherlands. They looked at fragments of bone over the geriatric skeletal remains that they had there, and they could predict the age of a skeleton in geriatric skeletons to about 10 years with small fragments of bone.
That's incredible, considering that when we use morphoscopic traits to complete an osteobiographical report, generally it's much easier to age juvenile and young adult remains. As the individual's age increases, it becomes increasingly difficult to pinpoint. But with bone histology, you're saying that you can estimate age within 10 years, no matter how old they are, and just with a tiny fragment of bone.
Yeah, so just a couple of centimeters you can get. Obviously, this gets a lot more accurate the bigger your sample is, but yeah, so with the ethics behind it, you definitely have to weigh this up. Obviously, if you have a complete skeleton, a very important, the ethics may outweigh this, and you may not want to go the invasive route and then do a more osteobiographical sort of approach where you can still find a lot about the person and from their skeletal remains.
And besides the invasiveness of this methodology, what other ethical considerations should we be keeping in mind before we consider this?
Yeah, definitely. Also, access to technology is a big one. There has been some headway in this area, definitely through ANU and Justina Mishkovitz. She did in 2020. I participated in one of her workshops, which she did over Zoom.
Oh, amazing.
Yeah, so she sort of taught people how to read these histological markers and then how to use the technology behind it. And this was definitely very important when looking at access to technology, because there is a lot of machines and technology that you need.
Cost and resources.
Yeah, need to use this. Not everyone has access to that, obviously.
If I saw correctly, Justina's hosting another one of these talks soon, right?
Yeah, she is.
We'll have to jump on that.
Yes, definitely. I think it's late April, so that's exciting.
That's very exciting. Now, unfortunately, we've reached the end of our little chat about bones and bone histology. But now I want to take a leaf out of some of my favorite podcasts' playbooks and give you the platform to speak about anything you wish about the topic of archaeology or history for 1 minute. Now, this can be anything from something you're fascinated with or passionate about. You can have a rant or use the time to share something that you love. It's up to you. You ready?
Absolutely. So I'm going to talk about one of my very early interests in my degree, which is Mesoamerican skeletal remains, specifically the modifications that they do on the bone.
Alrighty. Well, the platform's yours whenever you're ready.
Yeah. So with Mesoamerican remains, and this is mostly common in the Mayans, they do a practice called head shaping. So from a very young age, so about a couple of months old, from newborn to a couple of months old, they shape and elongate the skull, and they do this a multitude of ways, but often with boards. And then this is very cool because it then also alters the osteons inside the bone as well, which there have been a couple of cool studies from that. And then from this head shaping, it's obviously a wide cultural practice, so it's very cool to see. You can see the changes in dynasties and changes throughout main periods based on how they shape their heads. And they also changed their teeth as well. So they would file down their teeth, or they would add gems or often like obsidian to their teeth. And this was seen as like a form of status, altering the bone in that way, which I think is really cool.
That is very cool. Thank you so much for that, Mia. That was such an enlightening 60-second talk. Unfortunately, that's all we've got time for today. But for our listeners, if you're interested in learning more about the case studies that we spoke about today, bone histology, or the effects of health and disease on bone, keep an eye out for our blog on our website. Mia, it was so great to chat with you today. Thank you so much for joining us.
It was a pleasure. Thank you so much.
Join us for our next episode on the archaeology of people with Ashley Passfield. Until then, be kind to yourself and others. I'm Crystal Miller, and this was After the Dig.
Wait, guys. You forgot something important. References. We strive to be factually accurate by citing peer-reviewed sources on this show. In this episode, we cited 9 references. If you want to learn more about bone histology, how it can be used to study the past, or to read these sources directly, please check out our blog at www.afterthedig.com. But don't take our word for it. Go see for yourself. Until next time.